Hello, class. This is our recorded lecture that'll finish up chapter 5 in our neuroscience text, the second part of synaptic transmission. So let's take a look here at this synaptic vesicle. Now, they aren't just empty, smooth bubbles. Their membranes are very crowded with lots of proteins that control every step of the vesicle cycle. And we'll get into this in greater detail in a separate recorded lecture, the mechanism of this. But some of these proteins are transporters that load the neurotransmitters into the vesicle, while others anchor vesicles near the presynaptic membrane, or others that form the snare complex that drives fusion and eventual excitosis of the neurotransmitter. And when calcium enters the presynaptic terminal, these proteins coordinate a rapid docking, priming, and fusion system of vesicles, ensuring that the neurotransmitter releases occurs within just milliseconds of an action potential arriving. And so I want to talk about some neurotoxins that affect this system of snare proteins, of the docking and fusing and exocytosis of neurotransmitter. First, botulinum toxin. This is one of the deadliest toxins known. It's produced by Clostridium botulinum. It cleaves snare proteins, preventing vesicle fusion, and without neurotransmitter release. So without the snare proteins, there is no vesicle fusion, and therefore is no neurotransmitter to be released into the synaptic cleft. So in this case, with botulinum, the motor neurons can't activate any muscles because the neurotransmitter acetylcholine can't be released. And this results in what's known as flaccid paralysis, no movement. As you can see in the picture of the young child above that can't even open his eyes. And in severe cases, about 5 to 10% of those who are infected can experience respiratory failure. So this is a toxin that is life-threatening. Now, this toxin is made up of two major protein structures. This and tetanus is very similar, as we'll see. We call these AB toxins. That means they're made of two functionally distinct parts. There's the A or alpha chain and the B or beta chain, also known as the heavy chain. And they work together to disrupt neurotransmission. First, the B or heavy chain. It's responsible for targeting and entry. It recognizes the specific receptors on the presynaptic terminal, and it binds tightly to the presynaptic membrane. Once it attaches, it helps the toxin enter the neuron through endocytosis, the opposite of exocytosis. This is when the membrane is pinching back the vesicles back inside the presynaptic neuron. So you can think of the B subunit as a delivery vehicle. It gets the toxin into the right cell type, and motor neurons for the botulinum toxin, and as we'll see, inhibitory interneurons for tetanus. And so without the B subunit, the toxin would never get inside of the presynaptic neuron. Now, once inside the AB, because of the heavy chain, the AB toxin is now inside of a vesicle, inside of the presynaptic neuron. And the A, or the light chain, becomes the active catalytic part. So once inside the neuron, it escapes the vesicle and it acts as what we call a zinc-dependent protease. So this is a protein that breaks down other proteins. Zinc-dependent means that it has to interact with zinc for it to function. And what it does is it cleaves snare proteins, the very proteins that are needed for vesicles to fuse with the presynaptic membrane. And so without that snare function, vesicles can't release neurotransmitters. This is so the A, or light chain, is the business end of the toxin. It's the part of the toxin that actually causes the paralysis. Right? So one portion is sort of the delivery vehicle, and the other one is the actual the protease that causes the paralysis. Now, interestingly, the same mechanism is used medically. I'm sure many of you have heard of Botox. Botox injections use the AB toxin from botulinum, but it's used for cosmetic surgery. They inject it deliberately to paralyze muscles in tiny locations, in tiny doses. And it, because of that relaxation of the muscles, it reduces wrinkles, or treat conditions like spasticity, so uncontrolled muscle contractions. Here's an image that looks similar to the AB toxin of botulinum. This is for tetanus, and it acts in a very similar way, in which the heavy chain is the delivery system, and the A light chain is the protease that causes paralysis. So the tetanus toxin, this is made of a toxin made from a bacteria, Clostridium tetani, and it also, like botulinum, cleaves the snare complex, but it targets inhibitory interneurons in the spinal cord. And so these are interneurons that are responsible for sending inhibitory signals to our efferent motor neurons. And so by removing the inhibitory control, the motor neurons become hyperactive. And that leads to rigid paralysis, painful spasms, and the classic arch posture called opisthotonus that you can see here in this 19th century painting by Sir Charles Bell of a soldier suffering from a tetanus toxin. And so the difference between botulinum and tetanus lies in the neurons that they affect. One causes paralysis, the other causes this painful, rigid paralysis caused by painful spasms. So the opposite of the flaccid paralysis, where the muscles aren't contracting. Here, the muscles are contracting hyperactively, so much so that they can't relax. So botulism silences motor neurons directly, while tetanus silences the inhibitors, making them more proactive. Now, in the third and last toxin that I want to mention is called letrodotoxin. This is from black widow spider venom. It works in a different way than botulinum and tetanus. It hijacks proteins like nerexin and synaptotagmin, and it essentially forces the vesicle fusion, even in the absence of calcium. So it kind of does the opposite of the other two that prevent fusion. This one facilitates it, and it results in uncontrolled neurotransmitter release. Victims experience severe muscle cramps, spasms, sometimes systematic effects like seizures, nausea, cardiovascular problems. And these toxins reveal just how essential precise vesicle release is for normal neuronal function. In addition to activating synaptotagmin, the toxin itself can form this tetramere complex. So the four proteins together. So this, if you take 4 individual molecules of this toxin, they'll form a pore-forming complex that will insert itself into the presynaptic membrane, and thus neurotransmitters can escape through this pore as well, causing the same effects. Now, let's zoom out from the molecular level to the level of the whole neuron. A single neuron may receive thousands of synaptic inputs. Some are excitatory, some of them are inhibitory, and together they must be integrated to decide whether the postsynaptic neuron fires its own action potential. This balancing act is always happening and very dynamic in every neuron of your brain. Now, a few terms that are important for understanding this process. Summation. This is the process by which the PSP, the postsynaptic potential of a neuron, changes based on the additive contributions of signals from multiple stimulus inputs. They can be spatial summation or repeated signals from one stimulus input, known as temporal summation. So let's take a look at examples of these. Spatial summation. This occurs when inputs from multiple presynaptic neurons combine. So you can imagine 3 excitatory neurons all synapsing on the same postsynaptic cell at the same time. So individually, each may not be strong enough to fire an action potential. They can't depolarize it to threshold. But together, they can push that postsynaptic cell over the threshold, stimulating an action potential. Now, the second type of summation is temporal summation. And this comes from rapid repeated firing by a single synaptic neuron. If the inputs arrive quickly enough, the postsynaptic potentials overlap and they add together. This shows how both the number of inputs and their timing influence neuronal decision-making, if you will. Now we have 2 different types of postsynaptic potentials that are of significance. First, excitatory postsynaptic potentials, or EPSPs. They increase the likelihood of a postsynaptic action potential occurring. Therefore, they depolarize the postsynaptic potential. So excitatory doesn't necessarily mean an action potential will fire. It just means that it depolarizes. If it doesn't depolarize the threshold, then an action potential won't fire. And then we have inhibitory postsynaptic potentials, IPSPs. They decrease the likelihood of a postsynaptic action potential occurring. Therefore, they must hyperpolarize the postsynaptic potential. Excitatory postsynaptic potentials, or EPSPs, they usually result from sodium entering the cell, as you might have predicted. Obviously depolarizing the membrane when that happens. This makes the postsynaptic neuron more likely to fire an action potential. Inhibitory postsynaptic potentials, or IPSPs, they do the opposite. They hyperpolarize the cell. In the spinal cord, IPSPs often open chloride channels, and chloride comes into the postsynaptic cell. But in the brain, they often open potassium channels, so potassium can leave the cell. But both have the same effect, hyperpolarizing the postsynaptic cell. So these mechanisms make it harder for the postsynaptic cell to reach threshold, whether it be chloride or potassium. So let's take a look here, a figure from your book, different summations of both excitatory and inhibitory inputs. So we can see here E1 and E2, both delivering an EPSP, excitatory postsynaptic potential, to that postsynaptic neuron in this case. Either of those on its own is excitatory, depolarizes the cell, but not till threshold. Now, if both of these were to fire at the same time, so this would be a spatial summation, they are additive, and both together are enough to push the postsynaptic cell's potential to threshold. Thus, an action potential is stimulated. Here, the red inhibitory one, or I guess that's an I. The inhibitory neuron labeled with an I gives a inhibitory postsynaptic potential. You can see here in the figure in red that it's slightly hyperpolarized when it fires. And we might summate both an inhibitory and excitatory together, and they can cancel out. You can see here the excitatory neuron gives a little stronger of a depolarization than the inhibitory signal. So there's a slight depolarization, but it's much smaller than if the excitatory neuron were firing on its own. And this inhibitory, if all three were to fire at the same time in this spatial summation, you would see potentially that inhibitory signal is enough to prevent the additive effects of the excitatories of triggering an action potential, like they did in the second example. That inhibitory response, if this were in the brain, opening potassium channels would be enough to hyperpolarize the membrane just enough to prevent the larger depolarization signal from reaching threshold. And so the point here is that excitatory and inhibitory inputs don't exist in isolation. They add together. If a neuron receives strong excitation, but equally strong inhibition, the signals cancel. If excitation outweighs inhibition, the cell may fire still. So this constant balancing of excitation and inhibition is what allows neurons to compute complex information, filter noise, and maintain stability. It's a very dynamic process that's always happening. Excitatory and inhibitory signals coming in. So finally, the location of a synapse matters. Inputs closer to the axon hillock, also known as the trigger zone, have more influence. And so, what do you think makes the trigger zone have a greater influence? Why is it more sensitive? I'll give you a moment, or you can pause to think about what you already know about neurons that might make the trigger zone at the axon hillock more sensitive and more likely to determine the postsynaptic response. It kind of has the last say, outweighing other incoming signals. This one has a greater proportion of influence on whether or not the postsynaptic neuron will fire an action potential or perhaps become hyperpolarized and not. Well, it's because that in this area there's a higher density of voltage-gated sodium channels at the trigger zone. So even strong excitatory inputs on distant dendrites may not reach threshold if inhibitory synapses are firing near the soma. This is why inhibitory interneurons often target the cell body. They can effectively sort of veto whether a postsynaptic neuron fires or not. They can have the last say. So in summary, the precision of molecular machinery makes vesicle release possible. Toxins demonstrate what happens when that machinery is disrupted. And neurons integrate thousands of excitatory and inhibitory inputs in both space and time to make the critical decision of whether to fire an action potential in the postsynaptic cell. Our next subject in Chapter 6 will be all about different types of neurotransmitters and their interactions with receptors.